In the 1970s, autism affected one child in 2000. Today, the CDC says it's 1 in 36. That's not better diagnosis. That's a 50-fold explosion in a single generation. Genes don't change that fast. Something in the environment is triggering this, and the trigger is hiding in the one place no one looked: the gut. We sterilize the biological factories that build a functioning mind. Here's the evidence that shattered the genetic theory. Identical twins, same DNA, yet one develops autism, the other doesn't. The difference? A single infection, antibiotics, and a gut ecosystem that never recovered. The critical window is 6 months to 2 years, when the brain frantically wires language and social connection, and when one specific molecule must flood the system. When that molecule vanishes, genes lock shut and the brain builds itself wrong. DNA isn't just code, it's a library where most books stay locked. To read a gene, the cell must unwind tightly coiled thread. Acetyl groups keep it loose. HDAC enzymes rip those tags off, coiling everything shut. Gut bacteria produce butyrate, which handcuffs the HDACs, keeping critical genes readable. No butyrate, genes for social circuits, language centers, and emotional regulation lock away permanently. The brain never receives its instructions. Butyrate production is a partnership. Bifidobacterium shreds dietary fiber into acetate and lactate. Specialists downstream convert those into butyrate. Both crews must be present. Antibiotics obliterate either crew and the assembly line stops. Modern infants start with a crippled workforce. Infants don't produce significant butyrate until solid food arrives, around 6 months. That's when specialist bacteria colonize and butyrate spikes fourfold, exactly when the brain constructs its most intricate social and language networks. Disrupt the gut during this brief window, and the brain never gets the signal to flip gene switches. Regressive autism, a child who babbles then stops, happens when butyrate factories fail to ignite. In 2013 and 2016, researchers examined post-mortem autistic brain tissue. Not the genetic code, but whether it could be read. The DNA was normal, no mutations. But thousands of genes for synapse communication were coiled so tight they were functionally invisible. The genes weren't broken, they were imprisoned. This is exactly what butyrate deficiency predicts. Stripped acetyl tags, DNA wound shut, brain unable to read its developmental instructions. 3 discoveries sealed it. Wild mice have 10,000 times more microbial diversity than lab mice. Sterile lab mice exhibit rodent autism, repetitive behaviors, social withdrawal, gut inflammation. When scientists transplanted wild microbiomes into sterile mice, behavior normalized within weeks. Their brains rewired, and the timing matches perfectly. Butyrate production spikes fourfold between 6 and 12 months, the exact window when autistic regression typically begins. The smoking gun is wholesale destruction of an ancient partnership at the precise moment the brain needs it most. We engineered this. C-sections bypass the birth canal, the infant's first bacterial inheritance. Formula lacks sugars that feed bifidobacterium. Antibiotics, given to half of newborns in some hospitals, wipe colonies before they establish. BPA leaches from bottles. PFAS contaminates breast milk. Herbicides, patented as antibiotics, sterilize soil bacteria. Diagnostic criteria haven't changed since 1994, yet autism triples. Aging parents and pollution don't explain the twin paradox, the 6-month window, or post-mortem gene silencing. We created a world hostile to microbes that co-evolved with us for 200,000 years. Here is the final proof. When autistic children spike fevers, many suddenly make eye contact, speak, connect with their parents. This isn't magic, it's molecular. Inside every cell, heat shock proteins normally guard a master switch, keeping DNA tightly coiled. But a fever pulls those guard dogs away to deal with heat-damaged proteins. The switch activates. It recruits the enzymes that uncoil DNA, the same uncoiling that butyrate normally provides. For a few hours, the locked genes spring open. The circuits fire, the child is there, fully present, and when the fever breaks, the DNA coils shut again. This proves the genes aren't broken, they're locked in a room. We hold the key. In fiber, in dirt, in the bacteria we spent a century trying to eradicate. The question is no longer whether autism is reversible. The question is whether we're brave enough to restore what we destroyed.